Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Chiral Chromatography explained. Table of Contents. Introduction, Principle, Components, Types, Procedure or Steps, Factors affecting chiral chromatography, Common products and manufacturers of chiral chromatography, Applications, Advantages, Limitations, Troubleshooting and Safety Considerations, Recent Advances and Innovations. Conclusion. Introduction to Chiral Chromatography. Chiral chromatography is a separation technique used to resolve enantiomers, which are chiral compounds that exist as non-superimposable mirror images of each other, and for this reason it is also known as enantioselective chromatography. Enantiomers can be separated using several methods, such as crystallization, capillary electrophoresis, CE, and chromatographic techniques. However, chromatography has become the most widely preferred and effective approach for chiral separation. This technique holds major importance in the pharmaceutical industry because many drugs are chiral in nature, and the chirality of a compound can significantly influence its safety, therapeutic activity, and overall efficacy. Within a biological system, individual enantiomers of the same chiral compound can exhibit different pharmacological behaviors and biological effects. Typically, one enantiomer is pharmacologically more active and is referred to as the eutamer, while the other enantiomer may be less active, inactive, or even toxic, and is known as the distomer. A classic and tragic example highlighting the importance of chiral separation is the thalidomide case. Thalidomide was a racemic drug that was first commercialized in the late 1950s as a sedative. The drug was also prescribed to pregnant women to relieve morning sickness, without sufficient understanding of its chiral nature and biological consequences. One enantiomer of thalidomide possesses the desired sedative effect, whereas the other enantiomer is a highly potent teratogen responsible for severe congenital birth defects. Due to the lack of proper research and safety testing, the use of thalidomide resulted in more than 10,000 children being born with serious physical deformities. Even administration of the supposedly safe enantioma proved dangerous because thalidomide undergoes racemization within the human body, converting one enantiomer into the other. Therefore, the separation and isolation of pure enantiomers are crucial to ensure the development of safe, effective, and reliable chiral compounds, particularly in pharmaceutical applications. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Principle of chiral chromatography. The principle of chiral chromatography is based on the separation of enantiomers through their different stereoselective interactions with a chiral selector. A chiral selector is a chiral substance that can be incorporated either into the stationary phase or added to the mobile phase of the chromatographic system. Each enantiomer interacts in a unique way with the chiral selector due to differences in spatial orientation, even though they have identical chemical composition. These unequal interactions result in different strengths of binding between each enantiomer and the chiral selector. As a consequence of these differential interactions, The enantiomers migrate through the chromatographic column at different rates. Because they move at different speeds, the enantiomers elute from the column at different retention times, leading to their effective separation. Enantiomer separation in chiral chromatography can be achieved using two main approaches: direct and indirect methods. In the direct separation method, a chiral selector is directly present in the stationary phase or the mobile phase, allowing enantiomers to be separated without chemical modification. In the indirect separation method, enantiomers are chemically derivatized to form diastereomeric pairs, which have different physical and chemical properties. These diastereomers, unlike enantiomers, can be separated easily using conventional chromatographic techniques due to differences in polarity, boiling point, or stability. Several chromatographic techniques are commonly employed for chiral separation, including gas chromatography, GC, thin layer chromatography, TLC, high-performance liquid chromatography, HPLC, and supercritical fluid chromatography, SFC. Components of chiral chromatography. 1. Chiral stationary phase, CSP. The chiral stationary phase is fixed inside the chromatographic column and is responsible for the selective interaction with chiral compounds, leading to enantiomer separation. CSPs differ based on their separation mechanisms and the type of chiral selector used, making them suitable for different classes of analytes. Common types of CSPs include Perkel-type CSPs, brush-type CSPs. These were the first commercially developed chiral stationary phases. They separate enantiomers through specific non-covalent interactions, such as hydrogen bonding, pi-pi stacking, and dipole-dipole interactions. Perkel-type CSPs are particularly effective for separating pi-acidic racemates, including derivatives of amines and amino alcohols. Later developments combined pi-acidic and pi-basic interaction sites to enhance enantioselectivity and separation efficiency. Commercial examples, such as Welk-01, are still available through Regis Technologies. Polysaccharide-based CSPs. These CSPs are derived from natural polymers, such as cellulose and amylose. They are the most widely used chiral stationary phases due to their broad applicability and high enantioselectivity. Polysaccharide-based CSPs are effective for separating a wide range of compounds, especially those containing amide groups, aromatic rings, hydroxyl groups, and amino functionalities. Protein-based CSPs. These CSPs utilize biological macromolecules, such as enzymes or plasma proteins, as chiral selectors. Common examples include bovine serum albumin, BSA, ovomucoid, and human serum albumin, HSA. They are particularly suitable for water-soluble and biologically active compounds. Chiral recognition in protein-based CSPs occurs through hydrogen bonding, pi-pi interactions, dipole-dipole forces, and ionic interactions. Cavity-based CSPs. These CSPs separate enantiomers through inclusion mechanisms, where the analyte molecule is selectively accommodated within a chiral cavity. Cyclodextrins, CDs, are the most commonly used cavity-based CSPs and are suitable for analytes such as hydrocarbons, sterols, phenol esters, and aromatic amines. Crown ethers are another type of cavity-based CSP and are especially effective for separating primary amines. Macrocyclic glycopeptide-based CSPs. These CSPs use antibiotics, such as vancomycin and teicoplanin, as chiral selectors. The glycopeptides are covalently bonded to silica using linkers like carboxylic acid, amine, or isocyanate groups to improve stability. Enantiomer separation occurs through multiple interaction mechanisms, including hydrogen bonding, pi-pi stacking, polar interactions, and ionic interactions with functional groups such as hydroxyl, amine, and carboxyl groups. 2. Mobile phase. The mobile phase is the solvent system that transports the analyte through the chromatographic column. Commonly used solvents include methanol, ethanol, and isopropanol, depending on the chromatographic technique and analyte properties. The mobile phase can be modified by adding chiral mobile phase additives, CMPAs, which act as chiral selectors dissolved directly in the mobile phase. CMPAs form temporary diastereomeric complexes with analyte enantiomers, allowing direct chiral separation even on achiral columns. Common types of CMPAs include ligand exchange CMPAs. These involve chiral metal complexes formed using transition metals and chiral ligands such as amino acids or their derivatives. They interact selectively with analytes containing functional groups like carboxyl, amino, or hydroxyl groups, enabling enantiomer separation. Macrocyclic antibiotics as CMPAs. Vancomycin is the most commonly used macrocyclic antibiotic CMPA. These glycopeptides possess a unique three-dimensional basket-like structure that allows multiple interaction sites for effective chiral recognition and separation. Cyclodextrins, CDs. Cyclodextrins are widely used CMPAs due to their ability to form host-guest inclusion complexes. Hydrophobic regions of the analyte fit into the hydrophobic cavity of the cyclodextrin, enabling stereoselective separation of enantiomers. 3. Derivatizing agents. Chiral derivatizing agents, CDAs, are used in indirect chiral chromatography methods. These agents chemically react with enantiomers to convert them into diastereomers, which are easier to separate using achiral columns. Commonly used CDAs include Marfe's reagent and Mosher's acid, both of which are widely applied in pharmaceutical and biochemical analysis. 4. Chromatographic system. The most commonly used chromatographic systems for chiral separation are high-performance liquid chromatography, HPLC, and supercritical fluid chromatography, SFC. These systems consist of essential components such as pumps to deliver the mobile phase, columns packed with stationary phases, and injection systems for sample introduction. Detection of separated enantiomers is carried out using detectors such as UV visible spectroscopy, mass spectrometry, circular dichroism, and fluorescence detectors. Download our microbiology study app made for researchers like you. Types of chiral chromatography. Two main types of chiral chromatography are commonly used for the separation of enantiomers. 1. Direct method. The direct method employs chiral selectors either in the stationary phase, known as chiral stationary phases, CSPs, or in the mobile phase as chiral mobile phase additives, CMPAs. Among these options, CSPs are more widely preferred because they are commercially available, easy to use, and compatible with a wide range of chiral compounds. This method does not involve any chemical modification of the analyte, allowing the original structure of the enantiomers to remain unchanged. Separation is achieved through stereoselective interactions between each enantiomer and the Chiral selector, resulting in different retention times and effective resolution of enantiomers. 2. Indirect method. The indirect method involves chemical derivatization of the chiral analyte using chiral derivatizing agents, CDAs, to convert enantiomers into diastereomers. These diastereomers are then separated using conventional chiral chromatographic columns. Separation in this method is based on differences in the physical and chemical properties of the formed diastereomers, such as polarity, size, or stability. The indirect method is particularly useful when a suitable chiral selector is not available for direct separation. However, this approach is more complex and time-consuming due to the derivatization step, and generally provides lower resolution compared to the direct method. Procedure or steps of chiral chromatography. The first step in chiral chromatography is the selection of an appropriate chromatographic method, which is chosen based on the chemical and physical properties of the sample or analyte. Commonly used chromatographic techniques for chiral separation include high-performance liquid chromatography, HPLC, supercritical fluid chromatography, SFC, capillary electrophoresis, CE, and gas chromatography, GC. Among these techniques, HPLC and SFC are the most widely used methods because of their high efficiency, versatility, and suitability for a wide range of chiral compounds. After selecting the chromatographic technique, a suitable chiral selector is carefully chosen, as it plays a critical role in determining the effectiveness of enantiomer separation. The selection of the chiral selector depends on the chosen method, as well as the chemical nature, polarity, and functional groups of the analyte. Based on these factors, the stationary phase and mobile phase are optimized to achieve maximum resolution between enantiomers. In the direct separation method, chiral stationary phases, CSPs, or chiral mobile phase additives, CMPAs, are used to enable direct interaction between enantiomers and the chiral selector. In the indirect separation method, chiral derivatizing agents, CDAs, are employed to chemically modify enantiomers into diastereomers prior to separation. The next step involves sample preparation, where the analyte is prepared in a form suitable for chromatographic analysis. The sample is dissolved in a compatible solvent that does not interfere with the chromatographic system or the chiral selector. For indirect methods, derivatization is performed during sample preparation to convert enantiomers into diastereomeric derivatives. Once the sample is properly prepared, it is injected into the chromatographic column for separation. During separation, the sample travels through the column, and the enantiomers interact differently with the chiral selector present in the stationary or mobile phase. These differential interactions result in different retention times for each enantiomer, allowing them to separate as they pass through the column. In the direct method, separation occurs due to differences in stereoselective interactions between each enantiomer and the chiral selector. In the indirect method, separation occurs because the formed diastereomers possess different physical or chemical properties, such as polarity or stability. After separation, the eluted enantiomers are detected using suitable detectors, such as UV, fluorescence, or mass spectrometry, depending on the method used. The detector generates a chromatogram in which individual peaks correspond to each separated enantiomer. The retention times and peak characteristics of the enantiomers are analyzed and compared with known standards or reference values. to accurately identify and quantify each enantiomer. Factors affecting chiral chromatography. The chemical properties of the analyte, such as the presence of specific functional groups, molecular size, shape, and overall stereochemistry, strongly influence how the enantiomers interact with the chiral selector and affect separation efficiency. The type of chiral stationary phase, CSP, used, as well as the underlying chiral recognition mechanism involved, plays a crucial role in determining the selectivity and resolution of enantiomer separation. The composition of the mobile phase is an important factor in chiral chromatography, as it directly affects the strength and nature of interactions between the analyte and the chiral selector. The use and type of chiral mobile phase additives, CMPAs, can significantly influence enantiomeric discrimination and retention behavior. Mobile phase parameters such as solvent type, solvent polarity, and pH can alter analyte ionization, hydrogen bonding, and other intermolecular interactions, thereby impacting chromatographic separation. Temperature is another critical factor affecting chiral separation, as it influences the kinetics and thermodynamics of analyte selector interactions. Lower temperatures often result in stronger and more selective interactions between enantiomers and the chiral selector, leading to improved resolution. The flow rate of the mobile phase also affects chromatographic performance, with higher flow rates generally reducing resolution due to decreased interaction time between the enantiomers and the chiral selector. Common products and manufacturers of chiral chromatography. Chiral columns, chiral stationary phases, CSPs, are widely used products in chiral chromatography and are specifically designed to achieve efficient enantiomer separation through stereoselective interactions. Commonly used chiral columns include Chiral Pack, Chiral Cell, Aztec, Cyclobond, Aztec Chirobiotic, Welk 01, Lux, and Chirex columns. These columns are manufactured by well-known analytical companies such as Dassault, Sigma Aldrich, Regis Technologies, and Phenomenex, which are recognized for producing high-quality and reliable chiral separation materials. Chiral Mobile. Phase, additives, CMPAs, are used to enhance chiral recognition when separation is performed without a chiral stationary phase or to improve selectivity. Common CMPAs include beta-cyclodextrin, hydroxypropyl, beta-cyclodextrin, vancomycin, and L-phenylalanine. These additives are widely supplied by manufacturers such as Sigma Aldrich and Thermo Fisher Scientific, which provide high-purity reagents suitable for analytical and preparative chiral chromatography. Chiral derivatizing agents, CDAs, are used in indirect chiral chromatography to convert enantiomers into diastereomers before separation. Frequently used CDAs include Marfay's reagent, also known as FDAA, 1-fluoro, 2,4-dinitrophenyl, 5L-alanine amide, and Mosher's acid, also called MTPA, methoxytrifluorophenylacetic acid. These derivatizing agents are commonly manufactured and distributed by Sigma Aldrich and Thermo Fisher Scientific, and are extensively used in pharmaceutical and biochemical analysis. Instruments used in chiral chromatography are essential for accurate separation, detection, and analysis of enantiomers. Commonly used instruments include the Agilent 1-2-6-0 Infinity System, Alliance HPLC System, SFC Prep 150, and the Nexera series. These advanced chromatographic systems are produced by leading instrument manufacturers such as Agilent Technologies, Waters Corporation, and Shimadzu, and are widely employed in research laboratories, pharmaceutical industries, and quality control settings. Applications of chiral chromatography. Chiral chromatography is extensively used in the pharmaceutical industry for chiral drug discovery, development, and enantiomeric purity testing to ensure the safety and efficacy of medications. It is applied in agriculture to study chiral pesticides, herbicides, and other agrochemicals, helping to evaluate their biological activity and ensuring safer use with reduced environmental and health risks. In environmental studies, chiral chromatography is used to detect, monitor, and help remove chiral pollutants, contributing to environmental protection and pollution control. It plays an important role in the quality control of food additives by analyzing chiral compounds, thereby ensuring product safety, regulatory compliance, and consistent quality. Chiral chromatography is also employed in the production and isolation of enantiomerically pure chemicals, which are required for pharmaceuticals, fine chemicals, and research applications. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Advantages of chiral chromatography. Chiral chromatography offers high selectivity and sensitivity, allowing accurate separation, identification, and quantification of individual enantiomers. The availability of a wide range of chiral stationary phases, CSPs, and chiral mobile phase additives, CMPAs, makes this technique versatile and applicable to many different types of chiral compounds. Many drugs and biologically active molecules currently in use or under development are chiral, making chiral chromatography an essential tool in modern pharmaceutical and chemical analysis. In direct chiral chromatography, the chemical structure of the analyte remains unchanged, ensuring that both enantiomers are preserved during separation. By enabling the identification of the therapeutically active enantiomer and the removal of inactive or harmful enantiomers, Chiral chromatography supports the development of safer, more effective pharmaceutical products. Limitations of chiral chromatography. Chiral chromatography can be costly because chiral columns and specialized reagents are often expensive, particularly when used for large-scale or preparative applications. Method development in chiral chromatography can be time-consuming, as optimization of the stationary phase, mobile phase composition, temperature, and flow rate is often required to achieve effective enantiomer separation. A single chiral stationary phase, CSP, cannot be universally applied to all chiral compounds, making it necessary to test multiple CSPs during method development to identify the most suitable one. Chiral chromatography may face difficulties when separating complex mixtures, such as samples containing multiple enantiomers or closely related stereoisomers, due to overlapping interactions and limited resolution. Troubleshooting and safety considerations. Common problems encountered in chiral chromatography include peak overlap, baseline instability, and signal noise, all of which can reduce the accuracy, resolution, and reliability of analytical results. Peak overlap occurs when enantiomers have very similar retention times, making it difficult to distinguish and quantify them accurately. This issue can be resolved by optimizing chromatographic conditions, such as adjusting the mobile phase composition, changing solvent ratios, modifying pH, altering temperature, or selecting a more suitable chiral stationary phase, CSP. Baseline instability and excessive signal noise are often caused by detector fluctuations, improper detector settings, or general instrument-related issues. These problems can be minimized by optimizing detector parameters, ensuring proper equilibration of the system, and performing regular instrument maintenance and calibration. Safety is an important consideration in chiral chromatography, as some solvents, reagents, and chiral selectors used in the process may be toxic, corrosive, or otherwise hazardous. Such chemicals should always be handled inside a fume hood to prevent inhalation of harmful vapors. Appropriate personal protective equipment, PPE, including lab coats, gloves, and safety goggles, should be worn at all times to minimize the risk of chemical exposure. Pressurized chromatographic instruments such as HPLC and GC systems must be operated with caution, as high pressures can pose safety risks if not properly managed. Regular inspection of tubing, fittings, and pressure limits, along with routine system checks, is essential to ensure pressure safety and prevent equipment failure. Recent advances and innovations. Novel chiral stationary phases, CSPs, are continuously being developed, including zwitterionic materials that offer enhanced chiral recognition due to the presence of both positive and negative charges within the same selector. Immobilized CSPs and hybrid CSPs have also been introduced, providing improved chemical stability, longer column lifetimes, and better selectivity compared to traditional coated phases. Multidimensional chromatographic techniques, such as two-dimensional liquid chromatography, 2DLC, have significantly improved the separation of complex chiral compounds by combining different separation mechanisms in a single analytical workflow. Automation and high-throughput screening approaches have accelerated method development in chiral chromatography, reducing analysis time and increasing overall efficiency and reproducibility. The integration of chromatography with advanced analytical techniques such as mass spectrometry, MS, and nuclear magnetic resonance, NMR, has enhanced chiral analysis by improving detection sensitivity, structural identification, and confirmation of enantiomeric purity. Green chromatography approaches are gaining importance, with supercritical fluid chromatography, SFC, becoming more widely used due to its reduced solvent consumption, faster analysis times, and lower environmental impact compared to conventional liquid chromatography methods. Conclusion. Chiral chromatography is an essential technique for the separation and analysis of enantiomers and plays a vital role across multiple fields, including pharmaceuticals, environmental monitoring, and food safety. In pharmaceutical research and drug development, it ensures the safety, efficacy, and quality of chiral drugs by enabling accurate enantiomeric separation and analysis. The technique also supports environmental monitoring by allowing the detection and study of chiral pollutants, contributing to better environmental protection and risk assessment. In food safety and quality control, chiral chromatography helps ensure the purity and safety of chiral food additives and related compounds. Overall, Chiral chromatography improves the production and isolation of enantiomerically pure compounds required for research, industry, and therapeutic applications. Continuous advancements in chiral selectors and chromatographic techniques, particularly methods such as high-performance liquid chromatography, HPLC, have significantly enhanced chiral separation efficiency and expanded the scope of its applications. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!